Good afternoon. Welcome to Dr. B.R. Ambedkar Open University. And in today's class, we'll be dealing with organophosphorus insecticides. Earlier, we have studied 6 chapters, and this is the 7th chapter in insecticidal toxicology. So we'll see the chemistry, classification, mode of action, metabolism, and the toxicity of organophosphorus insecticides, along with carbamates and pyrethroids. So let me share the screen now. Yes. So in unit 7, we have organophosphorus insecticides. Basically, coming to the classification of pesticides, so pesticides are classified into 5 major categories based on their chemical structure and target: herbicides, fungicides, insecticides, rodenticides, and fumigants. And we are dealing with insecticides in that natural and synthetic ones are present, where natural ones are made up of plant-based ones and mineral oils, whereas synthetic ones are organic or inorganic. Coming to the organic one, there are organochlorines, which we have studied in our earlier class, that is unit 6. We have dealt with organochlorines, organophosphates, which we'll be dealing today now, carbamates, pyrethroids, all the three will be dealing now, and neonicotinoids that we'll deal tomorrow. So pyrethroids are based on their Halpasian group. They're categorized into type 1 and type 2. Type 1 is the absence of Halpasian group, and type 2 is the presence of Halpasian group. And this is the general diagrammatic representation of two neurons and an intervening synapse showing the sites of action of the most commonly used classes of insecticides. So we know that majority of the insecticides, they target the they are neurotoxic and they target the nervous system, right? So here you see DTT and pyrethroids, how they are affecting the sodium channels, and organophosphates and carbamates, they act on acetylcholinesterase, and nicotine and neonicotinoids, they act on nicotine acetylcholine receptors. Cyclodines, ivermectins, and piperonyls, they act on the chloride channels, and then they inhibit, they cause the toxicity. So this is how, so majority of the insecticides, they target on each of these receptors, and then they show their toxicity potential. And coming to the organophosphorus insecticides here, so there are more than 100 organophosphates which have been commercialized all over the world, and emphasis is given to the compounds which are now being used in India. The organophosphorus compounds, they were first developed by Gerhard Schroeder in Germany during the late 1930s and are one of the largest group of insecticides in use today. So majorly, they have two distinct features like first one is their acute toxicants of high level of activity, and secondly, they are chemically unstable. And here we are showing the general structure of the organophosphates, where R is the methyl or ethyl group, and O is the sulfur or oxygen group there. And X can vary a great deal and is the acidic group that is especially subject to chemical attack because of the positive charge developing on the phosphorus atom. And chemical names of compounds formed from phosphoric acid have the ite ending on those from phosphoric acid. Phosphorus acid have ITE, and phosphoric acid has ATE ending. So here you can see PO3, which is from phosphorus acid, has phosphite, which is called phosphite, whereas from the phosphoric acid we can have PO4, that is called as phosphate. Okay, so phosphite is rapidly and more effectively absorbed by the leaf foliage, whereas phosphate is poorly and less effectively absorbed by the foliage. So stabilized phosphites are able to penetrate foliage more effectively than phosphates. And coming to thiophosphate, so here when there is a sulfur atom attached to a phosphorus in the molecule, the chemical is designated by adding thiophosphate and the position of S atom is indicated. However, must be noted here that some authors do not distinguish a thionosulfur from thiol sulfur. So compounds in which one nitrogen atom is attached to phosphorus are called phosphoroamidates. And those in which two nitrogen atoms are attached are called phosphorodiamidates. Like down you can see. And pyrophosphates here, so derivatives of pyrophosphoric acid are called pyrophosphates. So these are certain chemical structural forms of this. and ethyl parathion and methyl parathion. So how to distinguish this? So this is the general structure of parathion. So the edges here, you have two overflankings over there, no, the to the oxygen atom. If that has an ethyl group, then it is called ethyl parathion. If a methyl group is present in that position, then it is called a methyl parathion. And coming to the symptoms in poisoning in vertebrates and in insects. So the mechanism of toxicity is basically to inhibition of the acetylcholinesterase, which leads to excess acetylcholine, overstimulating the nervous system, which further leads to paralysis and death. In humans and mammals, you see this. Whereas in insects, also the same thing. The inhibition of acetylcholinesterase causes overstimulation of the nervous system, leading to paralysis and death. Symptoms you can see in humans and mammals or vertebrates is nausea, vomiting, diarrhea, seizures, confusion, excessive salivation, and sweating. Whereas in insects, muscle paralysis, loss of coordination, death due to nervous system collapse has been seen. It's like more toxic in case of insects here. Examples of certain organophosphorus pesticides are chlorpyrifos, diazinon, malathion, and parathion, and their toxicity and symptoms vary based on their chemical structure. Toxicity to target is highly toxic to vertebrates, especially in large quantities of prolonged exposure, and effective against a wide range of insects, especially pests. Resistant development, yes, vertebrates do not develop any resistance, but repeated exposure can lead to increased sensitivity or chronic toxicity. However, in insects, they can develop resistance through metabolic changes or mutations in acetylcholinesterase. Coming to the treatment in vertebrates, atropine. So, atropine, pralidoxime, they can be given because atropine blocks the acetylcholine effects, and paradoxime, 2-PAM, they reactives the acetylcholinesterase. And supportive care, including ventilation, oxygen therapy, and seizure control, can also help. Whereas in insects, there is no treatment needed for insects because the purpose of using the organophosphorus here is to kill the insect. Environmental impact, yes, harmful to non-target species, including wildlife and aquatic organisms. Whereas in insects, impact on beneficial insects can also occur because we don't want beneficial insects to get affected. But still, this can also show its toxic effects on the beneficial insects. And pesticides can also affect natural predators and cause ecological imbalances. And coming to the mode of action here, so the organophosphorus compounds act by phosphorylating the cholinesterase, an enzyme that plays vital role in hydrolyzing acetylcholine. So complex physiological processes are involved in the transmission of the nerve impulse. So on receipt of the stimulus, this gradient is altered so that the inside becomes less negative and an action potential moves down the nerve. So when this potential reaches the nerve ending at the myoneural junction, acetylcholine is released, which diffuses through the synaptic gap between the nerve and the muscle and is absorbed onto the muscle ending. So this interaction of acetylcholine with the receptor end of the muscle gives rise to an action potential in the muscle that responds by contracting. So after its purpose is accomplished, the acetylcholine has to be hydrolyzed by acetylcholinesterase so that the muscle returns to its original state, ready to respond to future stimulus. That can be seen in this. And here, this is like more detail, like how this substrate and enzyme they act in the active site. What do they do? The catalytic and anionic site can be seen here. So the reaction between acetylcholine and cholinesterase takes place in 3 stages and can be represented by the following. At the first stage, it is an equilibrium between the enzyme and its substrate on the one hand and a complex of the two on the other. So the complex yields choline and acetylated enzyme in the second stage. The final stage is the deacetylation reaction in which the acetylated enzyme is hydrolyzed to give the free enzyme and acetic acid. So in the living organisms, these reactions proceed at a fast rate so that there is no accumulation of acetylcholine across the synapse or at the neuromuscular junction. The choline portion that is removed and the acetic acid combines again to form a new acetylcholine and the cycle is repeated. So coming to the inhibition of an esterase enzyme by organophosphorus compounds, here you can see basically the organophosphorus compounds react with acetylcholinesterase in the same manner as the normal substrate acetylcholine does. So in the first step, the enzymes form a reversible complex which is called Micadis complex with the organophosphorus compound. Then the complex breaks down, whereby a portion of the organophosphorus compound, the leaving group, is removed and the enzyme is phosphorylated or inhibited. So, in the last step, hydrolysis takes place and the acetylcholinesterase enzyme is liberated along with a phosphate residue, such as dimethyl phosphoric acid, so which is detached from the enzyme. In the hydrolysis step is much slower in comparison with that of the normal substrate, acetylcholine. As a result, the enzyme is not capable of dealing efficiently with bursts of acetylcholine entering the synapse as a result of nervous activity. So acetylcholine accumulates, resulting in the disruption of the nerve function. So excess of accumulation can be seen in this. The active center of the enzyme contains a negatively charged anionic site which binds the trimethylammonium group and a relatively non-specific esterate site which catalyzes the hydrolysis of the ester linkage. So this is a simplified explanation. In fact, acetylcholinesterase occurs with multiple forms and the inhibition of this enzyme may be a complex phenomenon involving several steps. And aging can also be seen here. So aging of the inhibited cholinesterase, where the rate of the recovery of cholinesterase inhibited by organophosphorus compounds increasingly reduces the time or contact becomes longer. So this is known as aging. What the organophosphorus compounds increasingly reduces as the time or contact becomes longer, so they become aged. You can call them like aging. So aging occurs since the alkyl group is removed from the organophosphorus portion of the molecule, which makes it make thus making the inhibited enzyme like more stable. Coming to the metabolism of organophosphorus insecticides here. um, Yeah, the metabolic rate is an important factor to affect the mammalian toxicity, insecticidal efficacy, persistency, selective toxicity, and resistance. So, organophosphorus esters interact with two major groups of enzyme systems, the microsomal oxidative state system and the non-oxidative enzymes such as esterases. So, the microsomal oxidative system in mammals is frequently localized in the microsomal fraction of the liver, while in insects, such enzymes are located mostly in the fat body. So the microsomal enzymes, they catalyze the conversion of molecular oxygen from air to an active form of oxygen. So this oxygen forms a complex with cytochrome P450 and thus brings about oxidative reactions. So the non-oxidative enzymes are of several types. All those enzymes that hydrolyze any phosphorus ester or anhydride bond are called phosphatases. And the enzyme can either activate or detoxify the organophosphorus insecticides. Coming to the activation reaction here. So these reactions are mostly catalyzed by mixed functional oxidases. Oxidation reactions catalyzed by these enzymes generally increase the toxicity. And we can see the desulfuration, oxidative desulfuration happening here. So one of the commonest reaction is the conversion of PS to PO. So here you can see the oxygen group being replaced. So this change generally results in an increase in the toxicity to the organism because the oxygen analogs are better inhibitors of target enzymes than the parent compounds. So the in vitro conversion of parathion to paraoxon, okay, has been demonstrated in both insects and mammals. So desulfuration also occurs in several organophosphorus insecticides, including dimethoate, phenyltrethion, and malathion as well. So later you can see the hydrolysis reaction, hydroxylation of N-methyl group followed by the dealkylation can be also seen here. So in the case of phosphoramidates, there is a hydroxylation of one of the N-methyl groups. And thioether oxidation to sulfoxides or sulfones is also seen. The divalent sulfur in thioether containing compounds provide an additional site for the oxidation to sulfoxide and sulfone derivatives. There is a rapid conversion to sulfoxide followed by a slow conversion to sulfone, and side group oxidation also happens, and degradation reactions where the enzymatic hydrolysis of an organophosphorus compound is the most important detoxification reaction, and the degradation of almost all organophosphorus compounds occur primarily by hydrolysis, and the action of both phosphatases on the anhydride linkage is of prime importance, followed by OD alkylation, and oxidative OD alkylation can also be seen. Dialkyl phosphates were further formed, which will be excreted through the urine. So this here, metabolism of dialkyl aryl phosphorothioates is given. First, dealkylation takes place, where the removal of the alkyl groups by cytochrome P450 of enzymes happens. Then, oxidative dealkylation comes, where the cytochrome P450 enzymes they add oxygen to the alkyl group, making it more hydrophilic. So, carboxyl esterase activity happens later, where the hydrolysis of the ester bonds happens, leading to less toxic or more water-soluble metabolites. So carboxyamidase activity can be followed later, which involves hydrolysis of amide groups or amide bonds if present to further detoxify the compound. So esterase hydrolysis, which is an esterase hydrolyze an ethyl ester to form an alcohol and a phosphoric acid derivative. And the last is isomerization, where isomerization might change the toxicity profile of the compound or could affect its biological activity. And in some cases, it may result in less toxic isomer, which may be more readily excreted from the body. So that is the metabolism. And coming to the treatment, so once the poisoning happens, so how do you treat it? So in serious cases of poisoning by organophosphorus compounds, so the order of treatment should be like this, where the artificial respiration is preferably by mechanical means, and inject 2 to 4 mg atropine sulfate intravenously and repeated at 5 to 10 minutes so intervals until signs of atropinization appear. In not serious cases, inject 1 to 2 mg of atropine sulfate if symptoms appear and repeated doses as necessary. So, inject pralidoxime chloride to PAM slowly intravenously if the patient fails to respond satisfactorily to atropine. Then, decontamination of the skin, stomach, and eye as indicated. And symptomatic treatment, example, inhalation of oxygen that can also help. And note timings of pralidoxime administration is most effective if given early after exposure before the organophosphates age and binds irreversibly to the acetylcholinesterase enzyme. We have studied the aging, right? So before that happens only, we have to give. And the higher doses of atropine in several cases, much larger doses of atropine up to 100 mg or more may be required to counteract the effects of acetylcholine overload. And this combination of atropine to block acetylcholine action and pralidoxime to generate acetylcholinesterase is the cornerstone of the effective management in organophosphatase poisoning. So some sort of model questions that you can expect from organophosphorus insecticides are to describe the mode of action of organophosphorus insecticides and what procedures should be adopted to treat the OP poisoning, and how does the activation of OPs takes place during their metabolism, and what are the different reactions that bring about the degradation of organophosphorus insecticide. So these are general questions that you might get from this unit, organophosphorates. So coming to the eighth unit, that is carbamide insecticides. So where we have, I've already told you, like today we'll be dealing with organophosphorus, carbamates, and pyrethroids. So coming to the carbamide insecticides, so in this chapter, we'll see the sequence of development of modern carbamate insecticides. It also provides information of the mode of action of carbamates and how does it differ from the organophosphorus. So the carbamate insecticides exist as esters of carbamic acid, typically having some kind of aryl ring substitute as the leaving group. And these compounds are more soluble in organic solvents, and other carbamates are more aliphatic in nature and may possess sufficient miscibility with water to act as effective plant systemic insecticides. So this is the in the carbamate you can see this is the general structure of the carbamate where R1, R2, R3 are either hydrogen, methyl, ethyl propyl or other short chain alkyls there. R3 is either phenol, naphthalene or other cyclic hydrocarbon ring. For example, if R1 is mm, hydrogen, R2 is methyl, and R3 is a phenyl ring. It is a phenyl carbamate. So you can call it as phenyl carbamate. So this is how you nomenclature is done there. So propagure, or when it is replaced with a naphthyl group, it is a naphthyl carbamate, like carbaryl. Naphthyl carbamates, example, carbaryl. Uh, During the same time, meiotic and cholinergic properties of physostigmine were discovered. The molecular structure was established. Physostigmine is derived from the colubrid beans, Physostigma venenosum, in witchcrafts. Uh, so a year later, the alkaloids were isolated, purified, and renamed as physostigmine. So this is the structure of it. Neostigmine is also the other compound of carbamate. Dimethyl is the other compound, and they suggested three major requirements for optimum contact insecticidal activity of a carbamate molecule, where it should be non-polar to be able to penetrate to the site of action in the insect nerve, and it should be structurally complementary to the active surface of cholinesterase, and it should possess sufficient stability to multifunctional oxidase detoxification. And here comes the mode of action of the carbamates. You can see uh, carbamate insecticides like the organophosphorus compounds, they inhibit acetylcholinesterase enzyme by acting as a substrate for the enzyme and resulting in the accumulation of acetylcholine in the nerve synapse and causing a disruption of nerve function. So when a carbamate insecticide enters the synapse, it competes with acetylcholine for the active site on the enzyme. So this you can see here like acetylcholine is there. So there will be an active site. So it competes with the acetylcholine. This carbamate competes with the acetylcholine for the active site on the enzyme. And the insecticide then binds to the enzyme and forms a complex with it. And this complex is reversible and the enzyme could be dissociated from the carbamate. So provided, however, in most cases, the reaction proceeds to step 2 where the complex decomposes into a stable carbamylated enzyme and a leaving group. Finally, the carbamylated enzyme is hydrolyzed to generate the free enzyme and methyl carbamic acid. So step 2 and 3 are much slower with carbamates than with acetylcholine but are faster than those with organophosphorus. So the carbamate reacts with a serine hydroxyl group within the enzyme site. So you can see the esteric side where you have serine, and you can see that the carbamate reacts with a serine hydroxyl group within the enzyme active site, resulting in carbamylation of this hydroxyl group and yielding a hydroxylated leaving group. So this process will inactivate the enzyme and blocks the degradation of the neurotransmitter acetylcholine. And here you can see the mechanism of action of acetylcholine, how it happens compared to the carbamates and organophosphates, where I have explained here, where the carbamates react with the serine hydroxyl groups with the enzyme active site, resulting in carbamylation of this hydroxyl group and yielding a hydroxylated leaving group. So they basically acetylcholine. So here you can see the schematic illustrating the reinteraction of acetylcholine, carbamate carbaryl, and the organophosphate fluorpyrifos oxon with the active site of acetylcholinesterase. The general rate of bond of ACH hydrolysis, everything can be seen here. So basically, acetylcholine is a neurotransmitter. It helps in the nerve function, whereas carbamates, they are neuroinhibitors. They are not neurotransmitters. They inhibit the acetylcholinesterase enzyme, therefore keeping the channels more open and not letting them shut down, so that the more stimulus upon the stimulus, more generation happens, and then eventually it leads to paralysis and death. And coming to the symptoms of toxicity in mammals and insects, so by using muscarinic and nicotinic receptors, so in muscarinic receptors, if they use, then it's excessive salivation, lacrimation, urination, defecation, and pupil constriction can be seen. In mammals, nicotinic receptors they use, then it's like muscle weakness, muscle twitching, muscle paralysis can be observed, and the LD50 values of carbaryl is 200 to 1000 mg per kg orally, and propofol is 50 to 500 mg. Methomyl is like too much toxic, like even 5 to 10 mg per kg can cause LD50. Aldicarb seems to be like very toxic, like zero point 0.1 to 1 mg per kg only it tolerates, and that too causes the LD50, lethal dose 50. In insects, motor dysfunction, twitching spasms, paralysis, uncontrolled movement, behavioral changes, disorientation, death, paralysis of essential muscles like wings, feeding muscles can be seen, and the LD50 value of in case of carbaryl is 1 to 10 mg per kg and methomyl is 0.2 to 10 mg per kg, whereas aldicarb is equally toxic to mammals and insects with 0.1 to 1 mg per kg. So carbamates methopropaction, yeah, as we've discussed earlier, the same we can see here. So the LD50 values for some widely used carbamates we have seen here. And this is the mode of entry of carbamate pesticides into the environment. How they will get out of it can be seen here. This is a picture that I've got from one of the publications, and all the publications, whatever the pictures I collect, I give their DOI number down, the journal. So if at all if you're interested and you want to learn in detail to enhance your knowledge, then you can directly click on this, or else you can copy paste this in the uh, Google or search engine, then you can get the full length paper of it and you can read it. So carbamates, upon their application, they get distributed into the environment via air, water, soil, and then the removal and transformation, how it happens, like exposure to biota of humans by inhalation, photolysis, human photolysis, alkaline hydrolysis, and microbial degradation. So all this happens once the carbamate enters into the environment. On coming to the metabolism of the carbamates are rapidly and more or less completely degraded by animals, and there is a prompt excretion of the metabolites. So usually within 24 hours, almost all the administered dose is eliminated, about 80% by way of the urine, and the major portions of the remainder through exhalation or fecus. So in the case of lactating animals, between 0.1 and 1% is eliminated in milk as well. So residues in tissues, milk, and eggs of treated animals are very small and 24 to 48 hours after treatment. So continued feeding of small amounts of carbamate insecticides does not cause a continued corresponding increase of residues in milk. Whereas carbaryl is degraded to at least 13 ether-soluble metabolites by rat liver microsome, and 15 metabolites of carbaryls were found in chicken urine, and Zectran was metabolized to 17 products by human liver microsomes. So this is how the metabolism happens, and hydrolysis, oxidation, and conjugation are the three important steps, the principal mechanisms of detoxification of carbamates. Where hydrolysis, the enzymes involved are carboxylase esterases. They convert carbamate to hydrolyzed metabolites like 1-naphthol and methylamine, followed by oxidation. The enzymatic oxidation most intensively studied has to be microsomal mixed function oxidase system. Through the mediation of the enzyme cytochrome P450 is involved here, which produces oxidized metabolites, example hydroxyl carbaryl, making them more polar. Uh, Hydroxylation, dealkylation, deamination, desaturation, epoxidation, and N or S oxidation. So this all takes place in case of oxidation. Coming to conjugation, conjugation is a process whereby endogenous substrates as well as foreign compounds react with natural body components such as glucuronic acid in mammals or glucosides in insects, glycosides in plants, as well as with other tissue components, principally sulfate, phosphate, and amino acids. So basically the purpose of conjugation is to convert the metabolic product to water-soluble compounds in order to facilitate their excretion or storage. So in animals, excretion takes place via the urine or fecus, whereas in plants, conjugates tend to be stored as terminal metabolites in various tissues. So conjugation of UDP, glucuronyl transferase, glucuronyl transferase involves, and sulfur transferase are involved, where conjugates metabolites with the glucuronic acid or sulfate to form highly water-soluble compounds that are easily excreted. So in mammals, they may be metabolized carbamates more slowly, depending on the liver enzymes, which can influence the susceptibility to toxicity. In insects, some insects may metabolize carbamates more rapidly or have different enzymes involved which can alter their sensitivity to toxicity. So that is the metabolism of the carbamates. And structure activity correlations. Here now we can see in alkyl phenyl and alkoxyphenyl methyl carbamates are involved. So in alkyl substituted carbamates, the anticholinesterase activity is usually correlated with ring position, meta, ortho, or para. So it depends on the ring position. The activity increases progressively with the addition of methyl groups. However, in the case of isopropylphenyl methyl carbamate, the difference in the insecticidal activity between the meta and ortho isomer is small, whereas the para isomer is much less insecticidal. The meta isomer of propulsor is almost inactive. Here we can see structural features. They're like correlations. Alkyl phenyl methyl carbamates, though the effect on activity is like longer alkyl chains, increased lipophilicity, higher potency in insects, longer persistence, shorter chains, faster metabolism, and lower toxicity. Whereas in alkoxyphenyl methyl carbamates, electron donating group, example methoxy, they show decreased potency, and electron withdrawing groups, example nitro, halogens, they increased potency and toxicity. The substituted phenyl methyl carbamates. So here halogenation of chlorine, fluorine, bromine, or nitro groups, they increase the potency and toxicity due to increased electrophilicity. Electron donating groups like methoxy, they decrease the potency. Carbaryl and multi-ring carbamates, where the naphthalene ring increases lipophilicity, stability, and potency. Halogenation, halogenation on the naphthalene ring increases potency due to electron withdrawing effects. And acetyl and other substitutions at the carbamate nitrogen. So the acyl derivatives of insecticidal carbamates formed by the substitution of the proton of the N-methyl carbamates by various acyl groups were generally less toxic to mammals than the parent carbamates. And such compounds show substantially less anticholinesterase activity than the parent N-methyl compounds, but their insecticidal activity is highly variable. For example, M-isopropylphenyl methyl carbamate is insecticidally active, but its N-acetyl derivative is practically inactive. N-methylation. So, N-methylation increases toxicity by enhancing the lipophilicity and binding affinity, whereas N-methyl carbamyl oximes show marked resemblance to the carbamates of cyclic enols and phenols. Moreover, the electronic effect of substituents on acetylcholinesterase inhibiting activity is more obvious in the case of the former than in the later. So, increasing electronegativity of substituents in the second position, they generally results in a corresponding increase in anticholinesterase activity. So, alkyl thiol substituents affect the biological activity in two ways. They provide potential for self-oxidation, thereby increasing the polarity, and they also increase the affinity for acetylcholinesterase. So, carbamate toxicity and potency depends on the delicate balance of the functional group effects and electronic properties, lipophilicity, metabolic stability. Whereas substituents on the phenyl ring, carbamate nitrogen, and aromatic systems, they play a crucial role in modulating these effects. Coming to the selective toxicity of carbamates. So unlike the organophosphorus compounds that we have studied earlier in the 7th chapter, so the carbamate esters, they do not show a broad spectrum of insecticidal activity. Some of these compounds are among the most specific of all insecticidal chemicals, and the factors that influence the toxicity of carbamates are its chemical structure, physicochemical factors, and biophysical factors. Coming to the chemical structure, though a few carbamates like aldicarb and isolon are highly toxic to both insects and mammals, others like carbaryl show different toxicity to insects and vertebrates. Even among insects and other arthropods, some of the carbamates show species specificity. For example, dimethane is highly selective for aphids, and dimethylen is an effective stomach poison for flies. So this is how it varies. So variations in the carbamate group, substituents, and multi-ring structure, they greatly influence enzyme binding, lipophilicity, and toxicity. Coming to physical chemical factors, so physical chemical factors also influence the toxicity of the carbamates. Strongly hydrophilic carbamates are slow in penetrating the cuticle, and hence they do not show contact toxicity. However, once they are able to gain access to the inside, so knockdown is quick and there is usually no recovery also. So strongly lipophilic carbamates, on the other hand, they penetrate easily, hence have a rapid knockdown effect that is followed by a degree of recovery dependent on the dose. And this recovery is probably due to the oxidation by the microsomes on lipid soluble materials. That can also happen. And a balance between lipophilic and hydrophilic properties and chemical structure is necessary in order to ensure penetration and to prevent degradation and excretion. So this balance possibly makes aldicab a successful pesticide. And coming to the biophysical factors, for substituted phosphates, a reasonably correlation exists between electrophilic properties of the phosphorus atom. In the case of phenyl diethyl phosphates, lipophilic bonding plays almost no role in the inhibition of acetylcholinesterase. But in case of carbamates, as much as 60% of the variation in acetylcholinesterase inhibition is accounted for by its lipophilic binding. So this is how the lipophilic binding is important in inducing toxicity. And differences in acetylcholine structure, metabolism, and blood-brain barrier permeability between species contributes to selectivity and persistence of carbamates in target organisms. It is interesting to note that resistance to carbamate insecticides develops more rapidly in insects against which the compound is inherently not very potent. So that is about the carbamates we can see. And some sort of questions that you can expect from carbamates chapter will be like how do carbamate insecticides undergo oxidative metabolism, and explain the mode of action of carbamates and compare it with that of organophosphorus insecticides, which can be asked as an a long question. It's a question. And what are the various factors that are responsible for the selective toxicity of carbamates insecticides? That is one more question that can come from this chapter. And write an essay on the development of carbamate insecticides. So that can be also the another questions that you can expect from this chapter of carbamates. And we'll be moving to the block 3 of a newer insecticides, in which we will see the synthetic pyrethroids. So earlier we have studied the botanical insecticides, in which we have dealt with pyrethroids, and in the newer insecticides portion, we will be seeing synthetic pyrethroids, where they are much similar to the pyrethroids, which was derived from the flowers of chrysanthemum. Yet their structure, the chemical structural difference, makes them uh, like a synthetic pyrethroid, the new insecticide. So pyrethroids are synthetic compounds based on natural pyrethrins as models, uh, whereas the photostable pyrethroids have proven to be broad spectrum insecticides, effective against a wide variety of insect pests and harmless to mammals and birds. They combine high insecticidal activity with suitable persistence. They are as much as 10 times more effective in the field than the most potent compounds of the other three principal groups of insecticides that we have studied earlier. They compare to organochlorines, organophosphates, and carbamates. They're 10 times more effective. So, originally, the pyrethroid that was derived from pyrethrum species, it was then discovered that chrysanthemum had much higher concentration of the insecticidal principles, and originally pyrethrum was marketed in the form of finely powdered dried flowers. Mm. A refined extract contains 25 to 30% pyrethrins, and general structure of pyrethrins you can see here, and the need for effective but safe and degradable insecticides was emphasized, and attention was focused again on the pyrethrins, natural pyrethrins as well. So development and for synthetic pyrethroids. So here you see the general structure of pyrethrins here, where ester linkage is present, alcohol moiety is present, and acidic moiety is also present. And in the nomenclature suggested for pyrethroids, the esters are designated as erythrins, and esters of chrysanthemic acid are designated as ones, and those of the pyretric acid as twos. For example, pyrethrin 1, which you can see here, is an ester of chrysanthemic acid, while jasmolin 2 is an ester of pyretric acid. Pyrethrins 1 and 2 are more potent insecticides than the cyanurins and jasmolins. The chrysanthemates are generally more potent for kill, and the pyrethrates are more potent for knockdown. So pyrethrates comes in the second group, and chrysanthemates in the first group. So they are more prone to kill, where you can see the knockdown process also in pyrethroids. So for this reason, pyrethrum extract has a broad spectrum of activity. Okay, so these are like pyrethrin 1, 2, cyanidin 1, 2, and jasmolin 1, 2. You can see here a structure of type 1 synthetic pyrethroids, where you see allanthrin, which comes in the synthetic pyrethroids. You see allanthrin. The first synthetic modification of the structure of pyrethrins mainly involved changes of the alcohol moiety, where you have seen earlier, like you have alcohol moiety, ester linkage, and acidic moiety, right? So the change in the alcohol moiety led to allanthrin, the first commercially available pyrethroid, and which is still in use today, was derived from pyrethrin 1 by shortening the pentadienyl side chain of the pyrethroline moiety. And bifenthrin is another pyrethroid, resmethrin, cispermethrin, cispinothrin, tefluthrin. So these are all the other compounds that you see. And coming here with this type 2 synthetic pyrethroids, where you get to see cypermethrin, which is frequently used. So when isobutyryl side chain was replaced by a dihaloine group, it results in a first photostable pyrethroid, permethrin. And the insecticidal activity was further enhanced by the incorporation of cyano group on the alpha carbon atom of the 3-phenoxybenzyl alcohol ester. Introduction of alpha cyano substituent in permethrin yielded cypermethrin. When esterized with dibromovinyl cyclopropane carboxylic acid, it resulted in an extremely effective molecule called delta methrin. So that is how. So changes in the structure, the smaller modifications will lead to newer compounds. That's what is we're dealing with here. Um, Hereditary. So in 1973, Sota et al. Altered the acid moiety of resmethrin by adding the thiol-lacto group to the acid chain that resulted in cadethrin. And this is the chemical structure of it. A very active knockdown agent. Cadethrin is a very active knockdown agent, and cyclic structures in the alcohol moiety appeared not to be essential for insecticidal activity as long as an unsaturated bond was present. The structure of several of these newly developed compounds is hardly comparable to that of the natural pyrethrins. So, as both the acid moiety and alcohol moiety were drastically changed, you cannot say that they are derived from the natural pyrethroid. So, their structure has immensely changed. Okay. So, examples of synthetic pyrethroids are permethrin, deltamethrin, cypermethrin. These are like most widely used, and their structural difference. So your reference has been given here. Now coming to the chemical structure and toxicity of the pyrethroids. So most pyrethroids have 1 to 3 chiral centers, which may be located at C1 and C3 of the cyclopropane ring. And pathology of pyrethroid toxicity is it's involved in immunotoxicity, carcinogenicity, epigenomic toxicity, reproductive toxicity, oxidative stress, neurotoxicity, and respiratory toxicity. And how it is affecting each and every of this is clearly mentioned here. And toxicity of synthetic pyrethroids and their effects on insects and mammals. Mm. So mechanism of toxicity, you can see here, basically the pyrethrins and most of the synthetic analogs are viscous liquids with low vapor pressure with high boiling point. So which prevents their environmental dispersion by air movements. So they are highly lipophilic, which favors their penetration in insects cuticle. So pyrethroids are also easily retained in the lipophilic cuticle of plant surfaces, minimizing translaminar action in leaves and systemic movements in plants. So the concentrations of pyrethroids in the plant cuticle will improve their rain fastness and makes them useful in application as contact insecticides as well. And the general effects on the insects you can see here with comparison to the vertebrates, where the mechanism of toxicity is inhibition of acetylcholinesterase, which leads to excess acetylcholine and overstimulating the nervous system, causing paralysis and death, which is same in case of insects as well. And symptoms in vertebrates will be like nausea, vomiting, diarrhea, muscle weakness, twitching, paralysis. Whereas in insects, it's like muscle paralysis, loss of coordination, mm, deaths due to nervous system collapse. And examples of certain pesticides of pyrethroids are chlorpyrifos, which is toxicity, and symptoms has been here given here, and diazinon, malathion, parathion, and their toxicity to humans and insects is shown here. Toxicity to target is like they're highly toxic to vertebrates, especially in large quantities or prolonged exposure. And they're effective against a wide range of insects, especially pests. Resistance development. So vertebrates do not develop resistance, but repeated exposure can lead to increased sensitivity or chronic toxicity. In case of insects, insects can develop resistance through metabolic changes or mutations in acetylcholinesterase. So treatment in vertebrates, you can use atropine, like we have discussed for organophosphorus. The same treatment will be involved here, like atropine, which blocks the acetylcholine effects. The same treatment is here because there is the same mechanism of toxicity. So that you have to remember here. And predoxime reactivates acetylcholinesterase and symptoms of so type 1 parathroids. The type 1 group includes the non-alpha cyanoparathroids. They were like too much of a toxic they cause. And in case of insects, no treatment needed for insects because the purpose of it is to be toxic to insects only. That's the reason they use this. And coming to the environmental impact, so we have seen how it will affect in various modes in our previous slide. And it is harmful to non-target species, including wildlife and aquatic organisms. And in case of insects, impact on beneficial insects can occur, and pesticides can also affect natural predators and cause ecological imbalances. So that is how we see. So in contrast, type 2 pyrethroids like delta methrin in mammals produce a distinctly different syndrome. Death is sometimes preceded by clonic seizures in insects, and the type 2 pyrethroids cause predominantly ataxia and incoordination as well. Whereas in humans, dermal exposure to either type of pyrethroid causes paresthesia, a tingling or burning sensation of the skin. But these effects is more intense for type 2 compounds, means they are like more toxic. So coming to the mode of action or the mechanism of action of pyrethroids, mm, here you mode of action, oxidation sites. Mm. Okay. So pyrethrines have a rapid paralytic reaction on insects. Even sublethal dosages results in a quick knockdown through the effect, though the effect is usually temporary and recovery is complete. Okay, so this total recovery indicates a reversible mechanism of action and also the ability of the insect to effectively metabolize small amounts of pyrethrines. So let me briefly explain you the mechanism of action or mode of action of pyrethroids. So basically what it happens is uh, we have been studying about the how it is going to affect the neural system, right? So in a similar way, most synthetic insecticides, so here we're talking about synthetic pyrethroids, so we have to deal with the most synthetic insecticides that target the nervous system proteins, like nervous system proteins. like pyrethroids like DDT. So these are like insecticides, synthetic insecticides. What they do is they bind to the voltage gated sodium channels and prevent its transition from an activated to an inactivated state. So they will prevent the transition from activated to inactivated state. So once it gets activated and it does its function, it has to become inactivate. But they will not let it become inactivate. They keep on doing the same job. As a result, the membrane of electrically excitable cells, they become persistently depolarized, and in the insect is paralyzed and dies quickly due to this, often exhibiting a knockdown response. So thus the first signs of the development of resistance to these compounds became known as knockdown resistance. So what will happen? Mm, when preventing the transition from activated, that is ion conducting, to an inactivated non-conducting state, results the membrane of electrically excitable cells become persistently depolarized, and the insect is paralyzed and dies quickly, often exhibiting a knockdown response. So that is what happens in case of action of the pyrethroids. So, and that is the diagrammatic representation that is involved there. And coming to the metabolism, so in general, pyrethroids are degraded by the hydrolytic and oxidative phosphorylation process, forming metabolites that are water soluble and thus can be conjugated and excreted. So the metabolites produced are of three major groups. One, those in which the ester bond is still intact, and second is the metabolites of acid moiety, and third are the metabolites of alcohol moiety. So this you can understand, right? Because earlier we have seen the structure of the pyrethroid, like how it is. It has one ester bond, it has a acid moiety, and it has alcohol moiety. So in order for its metabolism, you have to work on all these three things. Okay, so the latter two are produced as a result of ester cleavage. So in the case of animals, what happens is the exact nature and intensity of the degradation varies with the chemical and the species involved. And the sites of metabolism of some pyrethroids in mammals can also happen, where alethrin gets converted to tetramethrin, resmethrin, permethrin, cypermethrin, deltamethrin, and phenvalate, and each no, these are all the pyrethroids and they get affected at particular sites. So here you can see tetramethrin, desperethrin, phenolphthalein, permethrin. You see, no? So each target site will be different for each insecticide. Ester cleavage, you can see the ester linkage in pyrethroids is a vulnerable site in the molecule. So that is like more, but the ester hydrolysis is like more quickly done. It's like frequently attacked, or that is where you majorly attack and break and do the metabolism. So pyrethroids that are esters of primary alcohols are more susceptible to hydrolysis by esterases because they are the esters of primary alcohols. So esterases will come and act on the ester bond only. So while the ester bond remains intact in secondary alcohol esters, such as pyrethrins and elethrins, so non-esterase inhibitors such as organophosphates or alkyl carbamates increase the toxicity of primary alcohol esters, pyrethroids, indicating the importance of esterases in the metabolism of these pyrethroids. So extent of ester cleavage depends on the steric configuration. So you see here trans isomerase or cis isomerase. So it depends on the steric configuration of the acid moiety. The esters of trans chrysanthemic acid and trans isomers of those in which the isobutynyl group of the chrysanthemic acid moiety in primary alcohol esters is replaced by a dihalovinyl group, which you can see in permethrin, cypermethrin, and deltamethrin case. And that dihalovinyl group are hydrolyzed more rapidly than their cis forms, cis isomers. So trans isomers are hydrolyzed more rapidly than their cis isomers. And pyrethroids with the two gem dimethyl groups at the cyclopropane ring are metabolized slowly and at a rate similar to that of the cis chrysanthemic acid esters. So ester cleavage of the more stable cis chrysanthemates involves oxidation, which is the introduction of alpha-3 cyano group in 3-phenoxybenzyl alcohol esters. They were decreasing the rate of hydrolysis by esterases. The ester cleavage of these pyrethroids appears to take place to a large extent through oxidative process. So first the ester group will be broken as discussed now. Then the acid moieties will be broken and the oxidation steps depending on the chemical structures can also happen. So here what acid moiety has to be broken like the acid moiety of carotene is rapidly metabolized by oxidative cleavage of the sulfur containing ring, after which the liberated mercaptone is further degraded to give methyl sulfoxamide, methyl sulfone and sulfonic acid. And coming to alcohol moiety, so here the pentadienyl chain of the paratrenal moiety is oxidized to various diols. And the N-hydroxymethyl group of the alcohol metabolite found by the cleavage of the tetramethane is generally split off, and the benzyl ring is hydroxylated at the 2, 3, and 4 position. So here you see trans-tetramethrin, trans-bismethrin. So these are all the transforms, and deltamethrin, penvarilate, and penpropethrin were attacked in their particular sites, which indicates the oxidation sites depending on their chemical structure. So whatever the arrows that you see, those are the oxidation sites. When coming to the metabolism can vary based on the specific type of pyrethroid and the species that is involved and the functions. Okay. So in case of mammals, insects, plants, soil and water. So in case of mammals, the major route of metabolism in mammals is through ester cleavage by esterases. Esterases and oxidase attack as well as hydroxylation of the terminal aromatic ring. So this is the major route of metabolism in case of mammals. For example, cypermethrin, deltamethrin, penvalerate, and other stable pyrethroids are similarly metabolized. In case of mammals, they are similarly metabolized by the same mechanism with some modifications in the metabolic products. Mm. The low mammalian toxicity of the pyrethroids is due to the in vivo hydrolysis of the methoxycarbonyl group and the rapid oxidation of the isobutapyl moiety, which probably reduces the toxicity of the pyrethroids. And in case of insects, in general, it appears that metabolism of pyrethroids in insects is similar to that occurring in mammals, whereas with the slower rates of metabolism generated by insect enzymes, which are esterases and oxidases, than by those of the mammalian systems. Uh, Again, the trans isomers are more readily metabolized by esterases than the cis isomers, even in the case of insects. But there are exceptions, such as with the lacewing, in which the reverse is true. The importance of metabolism in the detoxification of pyrethroids by insects is evident from the increased efficacy of pyrethroids achieved by the mixing synergies. So specific esterase and oxidase inhibitors. So it depends on that. Coming to the plants, the initial metabolite process in plants are identical to those of mammals. That is, ester cleavage and hydroxylation reactions is like similar. However, oxidation of primary alcohol is a less important process in plants. The metabolites formed are usually conjugated with sugar or amino acids. And most pyrethroids are not translocated from the leaves or from the soil to other parts of the plant. In addition to metabolism, photo-induced reactions also occur on the surface of the treated plants. So in some cases, such as with the phenolphthalein, phototransformation is in fact the predominant degradation process. And ester cleavage, which is a major degradation process in plants, is photo-induced besides being a metabolic reaction. So in plants, the major process is a photo-induced phototransformation. That is what involved in plants. And coming to soil and water, so most of the stable pyrethroids insecticides undergo faster degradation in soils under aerobic conditions than under anaerobic conditions. So aerobic means in the presence of oxygen, and anaerobic conditions means without any oxygen, means in the absence of oxygen. So the rate of degradation varies from compound to compound and also with the type of soil. Phenolphthalein undergoes the degradation with the ester cleavage and the hydroxylation and oxidation reactions yielding metabolites, which are further converted to carbon dioxide. That is what happens. And in natural waters, permethrin is rapidly absorbed on the sediment and very little remains in the aqueous phase after 7 days. So phenvalate degradation under aerobic conditions is in the different soils lead to the formation of bound residues, and the evaluation of carbon dioxide are the major processes. So the stable pyrethroids are virtually immobile in soil, considering their higher lipophilic nature. Such immobility, coupled with the formation of bound residues and their subsequent complete degradation, suggests that the pyrethroids are unlikely to move from soil to any other part of the environment. They are like stick to the soil part itself. So these are the metabolism that can vary depending on the type of pyrethroids and the species that they are involved in. And the particular enzymes that are involved in pyrethroid degradation are esterases, cytochrome P450 oxidases, glutathione S transferases, and aminopeptidases, and UDP glucuranosyl transferase as well. So coming to esterases, so in mammals, mm, apart from the liver, so liver does the metabolism that we know, degradation, detoxification. So apart from the liver, this pyrethroid hydrolyzing esterases are also present in blood, kidney, lung, and stomach. So they are present in the liver, not only in the liver, but they are also present in blood, kidney, lungs, and stomach. And these enzymes are primarily found in the microsomal fraction, like very little. In the microsomal fractions, they are present. And rat liver microsomal esterases hydrolyzes bioresmethrin over 10 times faster than cismethrin, whereas no such isomeric specificity is exhibited by plasma esterases, indicating that there may be several different types of esterases that are responsible for the breakdown of pyrethroids. So we are studying few, but there may also be many more, like several different types of esterases which are responsible for the breakdown of pyrethroids. So based on their sensitivity to inhibition by the esterase inhibitors like paraoxone and alpha-naphthyl N-propyl carbamate, so different esterases could be distinguished for the hydrolysis of transpermethrin, cis-isomer and malathion respectively in mouse and rat liver microsomal preparations. So it was suggested that multiple esterases were present with different substrate specificities. So pyrethroid hydrolyzing activity in insects is widely distributed over various tissues. Mm, Highest is observed in cuticle, gut, and fat body for transpermethrin, and in cuticle and gut for the cis-isomer. Whereas no esterase activity was observed in the hemolymph of Spodoptera littorolus. And a study of kinetic properties, isomer specificity, and sensitivity to inhibition by carbamates reveal that species and tissue-dependent differences between esterases are present, and it is significant. And in cabbage looper larvae, more than one type of esterase is found in both the integument and the gut. So the cis and trans pyrethroid isomers are hydrolyzed by separate enzymes, and the rate of degradation reflects a difference in the activity of these different enzymes rather than a difference in the specificity for the cis trans pyrethroids of a single enzyme. So these are all about the esterases. And coming to the oxidases, so oxidases appear to vary between species because the microsomal monooxygenases are involved in the process of ester cleavage of pyrethroids. The extent of ester cleavage varies with pyrethroids and the species. For example, alethrine, when reacted with resistant housefly abdomen, it homogenates fortified with NADPH2, resulted in more than 10 metabolites, and in each metabolite, the ester linkage and the electrolyte moiety remained unchanged. So the major metabolic pathway was through hydroxylation of the trans-methyl group of the isobutynyl side chain in the acid moiety. A minor pathway that corresponds to the is involved in a similar series of reaction at the cis-methyl group In all probability, the actual detoxification steps were the ones that is succeeding and the formation of alcohols. At low enzyme rates, predominant metabolites were formed, but at high enzyme rates, they were mainly oxidized. Other unidentified metabolites of apolar nature were also found in small quantities. These were produced possibly by further oxidation and conjugation of the alcohol and acid moiety. Coming to the glutinous transferase, the type of enzyme involved is transferase, and it involved in conjugation reactions with the glutathione, increasing water solubility and excretion of pyrethroid metabolites. Examples are piperonyl butoxide and coming to aminopeptidases. So the type of enzyme is peptidase involved in the breakdown of peptide bonds, aiding in pyrethroid degradation. And UDP, glucuranosyl transferases, that type of enzyme is the transferases, and it catalyzes the conjugation of pyrethroid metabolites with glucuronic acid, facilitating the excretion. Okay, effects of synergesis on the enzymes. Um, Before we talk about resistance, we have to deal with the effects of synergesis on enzymes. So synergesis such as peperonyl butoxide, like we have seen here, peperonyl butoxide is an example of synergesis, where they were originally introduced to prolong the activity of unstable pyrethroids. So the basic intention of using this introduction of this PBO, that is piperonil butoxide, is to prolong the activity of unstable pyrethrum. And piperonal butoxide and also one more thing is like methylene dioxide phenyl derivative is also present. They are very much specific oxidase inhibitors and they are useful for increasing the toxicity. For instance, alethrin. Example, alethrin. So they what they do, the PBO and methylene dioxide phenyl, they increase the toxicity. So those synergies can increase the insecticidal activity of a pyrethroid. They are less effective on knockdown, indicating that pyrethroids generally bring about knockdown before significant elimination by metabolism has taken place. And coming to the resistance, so pyrethroid resistance in several species of arthropods is reported during the past decade, in spite of the fact that pyrethroids have been in use for a limited period of time in comparison to other synthetic insecticides. So cross resistance or multiple resistance conferred by DDT and organophosphates is the main mechanism by which this resistance has arisen. So resistance to pyrethroids was observed in the several species of insects which had a prior resistance to DDT. So some species of insects which are resistant to DDT, they tend to be resistant to pyrethroid as well. So pyrethroid selected strains of the housefly were cross-resistant to DDT. However, resistant to DDT does not have to be directly correlated with the decreased sensitivity to pyrethroids. Whereas organophosphate resistance was linked with the resistance to pyrethrins and pyrethroids in the flower beetle, and with increased susceptibility to fenvalet in the spider mite. So most knowledge of pyrethroid resistance has been derived from studies with the housefly. The reduced sensitivity may essentially be caused by majorly two factors: a decreased sensitivity of the nerve membrane and a decreased availability of pyrethroid at the primary target site, which may be mediated by several mechanisms like decreased cuticular penetration, faster degradation by metabolism, or storage in insensitive tissues, or increased elimination. So these are all can all leads to the resistance of this particular insecticide. So the absence of metabolic or penetration factors in resistant strains in several species of insects suggests nerve insensitivity as a mechanism for resistance, which is the most important of several pyrethroid resistance mechanisms. That knockdown resistance mechanism, which results in a broad cross resistance to pyrethroids, DDT, and DDT analogs for knockdown and kill, remains unaffected by synergesis. So it is located on chromosome 3 of the housefly genome and consists of one or more recessive factors. So this mechanism confers no cross resistance to organophosphates or carbamates. So the presence of the gene knockdown resistance or a similar nerve insensitivity is the most dreaded resistant mechanism. This gene, fortunately, is recessive in nature and can be managed by devising appropriate strategies to maintain heterozygosity in the knockdown resistant gene. So that is one sort of advantage in case of pyrethroids. So metabolic resistance involves the overproduction or enhanced activation of the enzymes that degrade or detoxify the pyrethroids. And target site resistance is the mutations in the voltage gated sodium channels. Target site for pyrethroids that reduce pyrethroid binding or efficacy and reduce the penetration by physical changes in the cuticle or cell membranes that reduce the ability of pyrethroids to penetrate the insect's body. And behavioral resistance involves changes in insects' behavior that reduce exposure to pyrethroids, like avoiding treated areas. If the insect does not come in contact with the treated area, then definitely there will not be any toxic effect. And most of the examples of insects that show metabolic resistance are mosquitoes, houseflies. Target site resistance are bed bugs and mosquitoes. So reduced penetration is mosquitoes and cockroaches. Behavioral resistance is cockroaches and mosquitoes. And we have already heard about the factors that influence the resistance. And these are certain factors that are driving the resistance, which we can commonly say is the overuse and misuse of pyrethroids in agriculture, public health, and household pest control. So the overuse should be limited because the more you use, the more chances of becoming resistant can be seen. And misuse is also not acceptable. The misuse can also lead to resistance because they get more frequently exposed to it and they build up resistance against it. Lack of resistance management strategies. Like you have to follow certain kinds of strategies to keep them like not being resistant to it, such as rotation with other insecticides. Instead of using one insecticide constantly and consecutively, you have to rotate with other insecticides as well, or using integrated pest management practices as well. And selection pressure due to frequent or improper application of pyrethroids, which favors the survival of resistant individuals. So, how you can manage or how you can combat this resistance is like you can use a rotation of insecticides because that is the major driving factor for resistance. One is by switching between insecticides with the different modes of action to avoid selection for resistance to any one class, and use of synergists like chemicals like piperonyl butoxide that inhibit the enzymes responsible for resistance, especially P450s and esterases, and integrated pest management, which combines chemical, biological, and cultural controls to reduce reliance on pyrethroids and mitigate resistance development. And also monitoring and surveillance is also important, which regular monitoring of the pest population to detect early signs of resistance and adapt control strategies accordingly are also beneficial. So this is how you deal with the factors that drive resistance, and you can easily combat the resistance by this management strategies. And the kinds of questions that you can expect from this chapter of pyrethroids is explain in detail the important milestones in the development of modern day stable synthetic pyrethroids is one question. Another is what do you understand by the type 1 and type 2 pyrethroids? And how do the pyrethroids get metabolized in general? And how do the synergists affect the effectiveness of pyrethroids? So these are the questions or the model examination questions that you can expect from this. So with this, we have covered three chapters of which we have dealt with the pyrethroids, carbamates, and organophosphates. And in tomorrow's class, we'll be dealing with molecules with different chemistry. Till now, we have seen the Things like we dealt with following a chemical basis of classification and all, but tomorrow we'll see with the molecules with different chemistry and their mode of action, their selectivity, their toxicity, like neonicotinoids. So which we have left. No, I told you in the first slide where we have shown ivermectins, abamectins. So all this we will be dealing tomorrow. Yeah. So we are done with the second, seventh, eighth, and ninth chapters. Thank you.